So now that we've practiced with some of the simpler Mendelian traits, there are other traits that we just generally call non-Mendelian traits. And so they're all going to be different, more complicated mechanisms where these genes are involved. One type of non-Mendelian trait is something we call polygenic inheritance. And this is the only one we're going to go into detail in for this class. When you go on and take biology, you'll do a little bit more, but we'll just focus here. Polygenic inheritance is going to be inheritance of traits that are controlled by multiple genes, where we have what we think of as kind of an additive or a cumulative effect. So on each one of these spots in your DNA, there's the possibility to code for activating something or not for activating something. And every time that something is activated, that's going to increase something. So if we look at like height as an example, maybe we have 10 different genes that control your height. And every copy of a gene that we have in an activated form, or you can think of it as like the dominant form, every copy like that is going to mean that you end up with a little bit more height. Examples of traits that behave this way in human beings would be things like height, hair color, eye color, skin color, and risk for certain diseases. One that comes up with this is called PKU. PKU is a disease that makes it so that a person cannot process a certain waste chemical that's in their body. We can't break it down so that it can be expelled. And so in people who have PKU, this chemical then builds up to toxic levels, and that can be deadly. So it causes brain damage when it builds up to high enough levels, which can be permanent brain damage. A lot of countries now, the standard practice is to test people for PKU, test babies for PKU when they're born. And so I remember this test with both my daughters. They right after they were born, like probably within 5 minutes of birth, they pricked their feet with a needle and put it on a little test strip to see if they tested positive for PKU. So let's switch back to height now. If we look at these two examples, we've got mom with this genotype that looks a little different than what we've seen before, and dad as well. The idea is that every time we see a capital letter, we're going to say that that's a gene that would increase your height. So for mom's genotype here, we only have one capital letter there, so mom's probably pretty short. Dad, though, has two capital letters, and so as a result, we would expect dad to be a little taller. Now, there's probably some difference in men to women on this as well, but anyway, just trying to keep it simple to begin with. In reality, human height is controlled by it's something like 10 or 12 different genes, so we're doing the really simplified version on this. So, let's say mom and dad, they produce offspring, and we're going to try and figure out how many of these little height markers the offspring are likely to get, or what are the different possibilities for this. So our next step then is to figure out the gametes that can be produced. And we did that before when we had just a single gene that we were dealing with, and now it's going to get a little bit more complicated. So every gamete that we produce is going to have one of these A traits and one of these B traits. So we have to think through what are the possible combinations for those different A's and B's. So for mom, we could have this first A and the first B. We could have the first A and the last B. We could have the second A and the first B, or the second A and the last B. And when we get into 3 gene traits, we end up with more different combinations. So that's where these get a little tricky. It's worth just writing down which different traits or which different gametes we have, so we don't have to do any extra work then. So mom can be big A and little b. Or she could be this A and, well, this B is also little, so that's still big A, little b. So the only other option then is the second A, which is a little a, and then either of these little b's would give the same result. So we're just going to have little a little b. So only 2 possible gametes from mom then. On dad we have a few more. We could do the first and the first, so big A, big A, big A, big B. We could do the first and the second, big A, little b. We could do the second and the first, little a, big B. Or we could do the second and the second, little a, little b. Oops. Okay, so let's set up our Punnett square then for the offspring on this. Usually we say the number of columns is going to be the same as the number of gametes from dad, and the number of rows is the same as the number of gametes from mom. So for dad, we have 4 different gametes, so I'll make this 4 columns wide and 2 rows high for mom. So 2 rows high and 1, 2, 3, 4 columns wide. And, I think I'm going to make this a little bit bigger so it'll show up on your screen a little bit better. There we go. So 4 wide by 2 high. And we'll put all of dad's gametes up top. Big A, big B, big A, little b, little a, big B, and little a, little b. And all of mom's on the side, big A, little b, or little a, little b. Let's just put a note. This is mom, and this is dad. Okay. Now when we write out the possible offspring genotypes, we just want to keep the A's together and the B's together, and then we'll always do the capital letters first if we have a heterogeneous mixture there. So first off, this spot right here, we have a big A from dad and a big A from mom, big A, and we have a big B from dad and a little b from mom, big B, little b. Here, we'll have a big A from dad, a big A from mom, a little b from dad, and a little b from mom. On this one, little a from dad, big A from mom, so we'll write the big letter first, big B for dad, little b for mom. And last one, little a for dad, big A from mom, little b for dad, little b for mom. Down here then, we've got big A from dad, little a from mom, big B from dad, little b from mom. We've got big A for dad, little a from mom, little b, little b. Here we've got little a, big B, little b. And last one, little a, little b, little b. Okay, so kind of a complicated picture on this one. We're going to sort these out based on how many capital letters we have. So it looks like we have some of these that have up to 3 capital letters down all the way to 0 capital letters. And these capital letters, we usually call those contributing factors. So 3 contributing factors. I see this one that has 3 capital letters or contributing factors, and it looks like that's the only one. A, big B, little b. For ones that have 2 contributing factors, oops, switch colors here. 2 contributing factors. Let's see, this one has 2, this one has 2, this one has 2, and that's it. Big A, little b, little b. We've got big A, big B, little b, and another big A, big B, little b. Alright, color here, one contributing factor. Got this one here. Crummy circle. Let's try that again. There we go. This one and this one. So got big A, little b, little b. Big A, little b, little b. Little a, big B, little b. And last one, 0 contributing factors. We've got this one, little a, little b, little b. Okay, so now if we total these all up, we've got 1 that has 3 contributing factors, 3 with 2 contributing factors, 3 with 1 contributing factor, and just 1 with no contributing factors at all. So this one up top here, this would be the tallest possible outcome with the 3 contributing factors. This is a little taller than average, this is a little shorter than average probably, and then this one would be a very short individual. So we could ask, like, what's the likelihood of having somebody who has 2 contributing factors? So let's try a problem like that. And for this, let's just use fractions instead of percentages, because sometimes the fractions will be kind of a pain, and that way we don't have to do any calculator stuff. So 2 contributing factors, well, we had 3 individuals like that. Out of our total of, it's 1, 2, 3, 4, 5, 6, 7, 8. So 3/8 of our offspring, expect there's a 3/8 chance that any one offspring would have just the 2 contributing factors. So for these multi-gene, these polygene traits, we have the possibility for a lot of different combinations. Don't forget about this step right here of going through the possible gametes. If we had left that off this time and just written out every single combination of those letters that we would have ended up with quite a bit longer chart. So it's worth your time to go through that extra step. It does save you time in the long run. There are now some practice problems on the next slide, so practice with those, and there are some solutions posted with each of those as well.